I need Good morning, everybody. Hope you're having a great day so far. Happy Tuesday. All right, if you haven't turned in the worksheet for protein synthesis code, please make sure that you do that. We're going to be doing a mutation lesson today. So in that module that says mutation lesson, please read that over if you already sent your worksheet in. Visual class. Okay. No, I thought I had already sent you the lesson, but I had not. So, here comes the professor. Okay. Good morning. Good morning. Good morning. Dang it. Almost everybody is here. In fact, everybody is here. Okay, good, good. All right, so before we get, so let me show you where we should be before we do that. All right, so you should have turned this one in already. That's the one I said you should have been able to finish it in one class period. I've done that. I've started a whole class and like, oh my gosh, it's not you. I fully understand. All right. I want to go over this for a minute because this part is we're going to repeat this activity in the lesson we're doing today. So it's important that you understand that the first strand is the DNA. And what we have to do is replicate that or translate it. Actually transcribe and then translate. And then the worksheet that we're going to have today, we're going to make sure that we have this handy so that we can then take the message that we've translated and remember that our RNA strand cannot have any T's in it. So every time we had an A, we had to put a U. And then we took that information and we went to the chart and we figured out what amino acid we're going to use to make that particular protein. So if you haven't turned that in, make sure that you do. All righty. So again, today we're going to use this chart. And the neat thing about this chart in your lesson is they have spelled out all the names of the amino acids for you. And there's one that let me make mine a little bigger first. Okay. There's one that on your chart looks like LL. It's not LL, it's IL, and it's isoleucine. Okay, so make sure that when you see that, you're not writing LL. It's IL. Okay, alrighty. And again, all of the names of all of the amino acids. And we have three that start with G, so we have glutamic acid is GAG. We've got glycine that can be written a couple of different ways. And we have glutamine, which is a C, it can also be written two ways, but it's CAG is one of them. Okay, so be careful with your G's and that one that starts with an I. Okay, when we abbreviate it. All right, so we are going to, let me come out of this a little. I think it's in your playlist already. If not, I have to go find it. Whoops. Okay. All right, so we're going to look at mutations today.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material, more specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random. But there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched. Insertion, which means an extra base or bases are.
Okay, I'm going to pause this just for a minute. Now, we are going to come back to all of these because you do need to know exactly what each of these are. but they are in the canvas. So like I said, we're going to come back to these. So do pay particular attention to these.
Added in. There is also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frameshift mutation. If you look at this frameshift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have half the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll give a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit 2 copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. While unfortunately there is not a cure for this disorder yet, the good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits 1 copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further reading suggestions in the description below. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so as you can see, that is already in your playlist. All right, so let's get back to the module. So we're going to just be worried about the first set of mutations. We're not going to do chromosomal mutations yet. we have a whole section on our next section is mitosis, meiosis, and we'll talk about those kinds of things there. But right now, we are just going to look at, so everybody be in Canvas, look at the lesson here, and we're going to look at types of mutations. This is very short. Go through that. Yes, you've heard of mutations. You just heard of mutations. And it was something that she said too, that they are random changes in the DNA. And because DNA gives instructions, pause right there for a minute, because the DNA is given instructions, if you have a random change in the DNA, then you're going to have a random change in the instructions. And the instructions are to go get a particular amino acid and make a protein. So if you change the instructions, you're going to go get a different amino acid and you're going to create a different kind of protein. Sometimes that's a good thing, because it might make the organism better. And sometimes it's a bad thing. It could either cause a disease or it could also be lethal. Okay. So anything that causes a mutation is called a mutagen. And that could be anything being exposed to ultraviolet radiation, some tars and tobaccos, or asbestos, for example. So they are random. You never know when you're going to get one. Okay. So there are different types of mutations. So basically this one just went through what the slideshow just showed you. And the video talked about gene mutations and chromosomal mutations. So we're going to just talk about gene mutations today. And we're going to actually do a worksheet that goes through how we get gene mutations. So do read these over. But you can have missense where it results in a different amino acids, but it may not have an effect. If you remember looking at the chart when we went through our little chart, some of the amino acids have 3 and 4 different ways that you can get that same amino acid. So if you change one letter, it doesn't have any effect at all because you're going to still get the same amino acid. Then you have nonsense. If you changed it, and it might create a stop where it shouldn't have a stop. Like technically the one on your worksheet had a stop, 2 stops, because we didn't really translate it back into transfer RNA, but that's okay. You just need to practice. We're going to do the same thing with the worksheet you're going to do today. So you create a protein that is non-functional. So again, when we talk about when we do chromosomes, we're going to talk about different diseases. All right, or silent. Okay, the base pair results in the same amino acid but no change. All right. What else? The video talked about frame shifts, whereas if you insert one or delete one, it creates, we're supposed to be reading them 3 at a time. So if we take one out, that changes our set of 3. If we stick one in, that also changes our set of 3. So that's a frame shift. Creates a different set of codes. So again, we're going to come into doing this later on when we do mitosis and meiosis. All right, so before we get ready to try to do all of this, I want you to shift pages. I want you to download this worksheet, and hopefully mine will download this time. And I'm going to go through some of it with you. So you need to stay with me. All right, mine keeps doing that, but hopefully yours won't. So I'm going to pull it up a different way. All right, so your worksheet should look like that. So let me know if you get it or if you had the same issue I just had. And again, you got to stay with me because we're going to work this together. All right. What I also want you to have is it's going to be real helpful. Let me see, because you can't see unless I have this in a certain place. It's going to be real helpful if you have this chart sitting somewhere off to the side, because you're going to need to refer to it. So there is one attached to this worksheet, but it's all the way at the bottom, and I don't want you to have to keep scrolling to the bottom to look for it. So if there was a way, like I tore mine off because I print mine. But if there's a way that you can have a double screen where you make the one that I sent you earlier visible, then that would be helpful to you. All right, so we have a cow and a human, and we are going to look at, and again, you're going to have to submit this, but I'm going to work on this with you. So make sure you're staying with me. All right, and again, it helps if you completed the last worksheet because it had a lot of points that we made on the last worksheet that go with this one. Okay, so what you have, and we're going to use this box to answer some of the other questions. So we got to fill this box in first before these two boxes before we go on to the other parts. All right, so below you have two partial sequences of DNA bases. Sequence 1 is for a human and sequence 2 is for a cow. Both humans and cows in this sequence are part of the instruction for controlling a bodily function. In this case, the sequence contains the gene for the protein insulin. So what they're trying to say is both cows and humans need insulin. Anybody diabetic in the class? Anybody got diabetes? Anybody got a family member that's got diabetes that needs insulin? Okay, and you're all very healthy. That's a good thing. All right, but we make insulin, and cows make insulin too, and their job is to regulate the
Um, I
question.
can't find the worksheet. Is it in the to-do list?
It's on the to-do list, and it should also, let me come out of mine for a second here. It's in the module. You might have to refresh because I think I just transferred it over there from the other. I thought I had it loaded already. Refresh your page if you don't have it. But it should be in the module right after the lesson on gene mutation lesson. So right after the mutation lesson, so if you the lesson we were just on, if you're in the module and you go, come on, go to the next page, it should be there. You got it? Like I said, you might have to refresh. Okay, good. All right. And for some crazy reason, when I tried to make mine pop up, it's not cooperating with me. So I'm going to look at this from my list there. All right, so again, insulin is necessary for the uptake of sugar in the blood. Without insulin, a person cannot digest sugars the same way other people, and they have the disease called diabetes. All right, so this incident is going to be a mutation that causes a disease. All right, so first we're going to look at what normal DNA looks like in a human and in a cow, or the normal amino acid sequence. So we're going to take each of these boxes and you're going to help me and talk to me. This is going to be an interactive lesson. So I'm hoping that you will practice how to do this. So the first row at the top is the DNA, and then we have to translate it. So we're going to create first a triplet code for the RNA. So I'll do the first one, and then somebody can tell me what the second one is. So if we have C, C, A, every time we have a C, we have a G. And so that's going to be G, G. And every time we have an A, it's not, if it was DNA, it would be T, but this is RNA we're making, so it's going to be U. So our first box is, and then you should be writing it down. The first box is GGU. And then we go to our chart and we figure out what is GGU. Now I'm going to scroll mine down. All right, so begin to practice using this chart. First letter is G, so that makes it this, let me make mine bigger. So there we go. All right, there you go. All right, so G is down here to the bottom. And then I go up to the top and find G again, because that's my second letter. And then I come down to the bottom because I know it's got to be in this bottom row. And I look for the U, and that's the first one. But in this case, so I see GGU, but in this case, all of them, there's four different ways to write glycine, but glycine is the amino acid we're looking for. So in the box where it says amino acid, you're going to write GLY. Okay. All right. Everybody with me? All right. So the next one is, see, this is what I was trying to keep you from having to do. All right. So the next one is TAG. Somebody sounds like they're ready to tell me what it is.
I was gonna say AUC, and then the amino acid is I.
ILE. Exactly. Perfect. Perfect. She said, thank you, Imani. She said AUC is our RNA triplet code. And we're going to go on our chart. And I'm going to run down here to the chart. And so I start with A is this one right in the third row. And U up here is the second letter. So that's going to be third row, first column. AUC is right here in the middle. And it's one of three different ways that you can write isoleucine, I-L-E. So I-L-E is going to go in your box for amino acid. Okay. All right. Now I'm not going to keep running back down here to the chart, but somebody else tell me what CAC is going to translate to. And it is nicer if you just unmute rather than try to type it in the chat. Come on, somebody, anybody. Follow Imani's lead. Or if you just must type it in the chat, type it in the chat. All right, I hope this silence means you're busy at work trying to figure it out. Let me see something. Can I make this? Do I like that? And I can scroll faster. Okay. All right, silent people. The next one, our CAC, is going to be GUG. So we come down to our chart and we do G, and then we come up here and do U, and then we come down and we look for, and technically you're going to the third side and you're looking for G again, which would bring you back over here to this box and GUG. And then again, this one for valine, there's four different ways to write it, but at the bottom here, we see our GUG. So that valine goes in the next box. All right, the next one is GTT. Anybody already did that one? Come on, folks, participate. Practice makes perfect. You got to do this. Got to do this. Don't wait for somebody to keep giving you the answers. Oh, wait a second. Hold on. You know what my problem is? My answer box is weird. Okay. All right. Okay. I see, Andrew, I see where you answered the other one too. All right, somebody else answered this one and no, it was not HIS. And folks, when you answer in the chat, please send it to everybody, not to me. Because we want everybody to see the answer. All right, so what is GTT? How do we translate GTT?
CAA.
Yes, indeed. You said it and Raymond agrees. And what is that going to? So C, and then A is our third one. And then we come down to our row here. And this one is going to our CAA is going to be GLN. Okay. CAA is going to be GLN. So that's going to go in that box. Very good. Very good. All right. Next one. Our ACA. Okay. Thank you. Hey, Jamarian. Yay. All right. UGU. And that's we go U up here at the top, and then U again. G is our fourth column. And we come down to our U, which is the first one at the top. So our amino acid is cysteine, CYS. All right, everybody getting the hang of this? Okay, next one is TGA, and that's going to translate to if you have to make a note to yourself on the side, make a little note on the side of who matches who. All right, that's okay. ACU, good, good. And then we go on our chart, and A is this third row, C is the second column, and we come down. ACU is THR, and then this is one of those, there's 4 ways to get that amino acid. Now, it's important that you pay attention to the fact that some of these, there's 4 ways to do it. Some of these, there's only 1 way to get it. There's 2 ways. That's important. It's going to be a question that's going to be asked later on. Okay, AGG is going to translate to AGG. Okay, UCC is this one. All right, U, C, and then C. Technically, like I said, you come across, you can shortcut that third one by just saying, if I know it's the second row and second column, then I go look and find the combination in the box and you'll find it. But technically, you're supposed to go the third letter, go across. And again, we have serine. Okay, UCC is serine. Again, 4 ways to get serine. And our last one is AAA, and it's going to translate to, I'm gonna help you with that one. It's gonna trend. Thank you, Amani. You. And it's going, so you, and the first row up here is you. So that's going to be PHE, and that goes in that box. All right, so you want to do the same thing with the cow. So that was the human segment. Now we want to do the same thing with the cow. So I'm gonna give you a few minutes to do the cow. Everybody work on your cow segment. And then I'm gonna come back to make sure to help you with the analysis. Now keep in mind, both of these are for insulin. So insulin in the human and insulin in the cow. So it's the same substance. But what we want to look at now is it made differently in a cow? All right, so take a few minutes to go ahead and fill in your RNA strand, then find your amino acids. And then the first thing they want you to do, I think I can do this, is look at both sets of DNA strands and circle on the DNA strand for the human and the DNA strand for the cow. Circle the ones that are different. Some of them are going to be the same in the sequence, and some will be different. And then after you circle them, tell me how many you found. Then you want to examine the amino acid rows and see how many times the amino acid is different. And you're going to highlight it. And then write down how many times you got to highlight. All right, so remember what I said about knowing these things in the box. So then question number 3 wants you to explain how you can have different codings and still get insulin. All right, so I'm gonna give you 10 minutes and then I'm gonna come back and ask you for your answers. In fact, I'm gonna stop sharing for a minute and do attendance. I'll be right back. Everybody needs to be working on this. Oh, I see a couple of screens where you're not working on it. Huh. How's it going? Anybody run into a problem? All right. I wish I could write on mine so that I could have one up, but hopefully you are staying on top of it and actually working on it in real time. In other words, use class time for classwork and not be wandering off somewhere else. All right. Hopefully you have completed your cow data. So now you want to look at the cow DNA strand. That's your top row on the cow and your top row on the human. And you want to go across and be comparing them as in sets. So you want to look at the first, like for example, the first one on the human is CCA and the first one on the cow is CCAs. Good. Second one on the human is TAG, but the second one on the cow is AAG. Circle. Okay. Because what we're doing with question number one, we are circling the DNAs that are different. So we're going to circle that second one. All right, the next one for the human is CAC, and for the cow, it's CAT. Different. Circle. All right, then we have GTT for both the cow and the human. We have ACA for both the cow and the human. We have ACG for both the cow and the human. We have TGA for the human, but CGA for the cow. Circle. Both top and bottom. All right. Next we have AGG for the human and AGG for the cow, same. And then we have lastly AAA for the human and GAC for the cow. Different. Circle. So how many circles do we have? Question number one, how many bases are different? No. We're just talking about the row. So we look on both rows. And how many times did I tell you to circle? You should only circle the ones that are different. So how many did we circle? The rest should be the same. And again, I'm talking about the DNA strand, just the DNA strand in both the cow and the human. Now, I don't mean all together. I mean, just if you were looking at sets, how many sets are different? You're not counting each one. Come on, folks. Y'all were supposed to be doing this with me. No. All right, let's go back over that again. All right, let me see. All right, if you look at this column right here, that was our first one that was different. So that's circle number 1. And again, I wish I could write on this. All right, so then the next one, they were different. So that was 1. Next set, they're different. You got CAC up here and CAT down there. That's 2. All right, these 2 are the same. These 2 are the same. And then we get over here, and these 2 are the same. And then we get here. This one and this one is different. So that's 3. All right, then these 2 are the same, and then these 2 are different. That's 4. Okay. Answer to number 1 is 4, and you should have 4 things circled. 4 on the top and 4 on the bottom. Close enough. But if you were doing this with DNA, close enough will give you a defect. So we don't want close enough. We got to be right. All righty. So we got 4 is the answer to number 1. And then again, you should have 4 circles on the top, 4 circles on the bottom. All right. The next one wants you to examine the amino acids that you produce. So we did the first one together. Hopefully you have filled in the second one, and you should have GLY for both of the first one and the second one. You should have PHE, right? UUC, All right, and you had for the top one, you had ILE. So that's different. Okay, that's different. So you want to highlight that. All right, the next one. You had Val at the top, and Val should be the one that you have at the bottom, even though you had a different way of finding it. You still have Val at the top and Val at the bottom, but it still made the same amino acid, so we're good. All right, the next one, you should have had GLN at the top, and you should have GLN at the bottom, so it's making the same amino acid. At the top, for human, I'm going to start human and cow. Human was CYS, and then CYS for the cow. The next one was CYS for the human, again, CYS for the cow. And then we're up to our TGA for the human, translated to thurine, which was THR, and our Cow translated should have retranslated to alanine. Okay, so we had G. We should have had G, Cu, and that's alanine, which is different than the thurine. Okay, our next box should have translated to serine, which is the same as the one at the top. And our last box should have translated to CUG, and CUG is leucine, and at the top it was UUU, and that was PHE. So that's different. So now how many different amino acids do we have? Okay. Now this time you weren't circling, you were highlighting. So how many highlights do you have? I, Imani, I think that your answer to this question, you got 3.
Are you asking like in sets of highlights or just each one highlighted?
It'll be a set. You can call it a set because we're looking at top and bottom. So it was the same thing with the other ones. Sets. So yes, 3 sets. If there are 3 different amino acids, there's 3 sets that are different. So yes. All right. Everybody got that? All right. Now, so we've clearly indicated that there's a difference between the DNA and the RNA. Why are they still making insulin? Why can they still make insulin? I'm going to give you an example. My daughter's name is Teresa. There are 3 different ways you can spell Teresa. Some people spell it with Theresa. Most people spell it Teresa. My daughter, because she's named after her father, who's Terrence, and had 2 R's in his name, so she has 2 R's in her name. So but we pronounce them all Teresa. So it's going to be the same thing with our insulin. There's several different ways we can arrange the amino acids and it still spell insulin. Okay, or still produce. In this case, we're going to say produce. Still produce insulin. All right, so I've just answered number 3 for you. So hopefully everybody's writing that in. So there's a difference in this DNA sequence for insulin, yet they make the same protein, insulin. And the answer is yes, they can be the same because there are different arrangements on the amino acids that still produce the same protein. All right, so now we're going to look at some of the types of mutations. And the first one we're going to look at is point mutation. So point mutation, like with the diabetes. So this is what we were looking at with our insulin, is the inability to break down sugars. Often people with diabetes have a defective DNA sequence that codes for making insulin protein. This mutation is called a point mutation. Now, you might want to make a side notes to yourself because you're going to have to match up the disease with the type of mutation. So you might want to keep a little side cheat sheet for yourself that says diabetes is point mutation. Okay. Test, test, test. Oh, excuse me. Excuse me. All right. Only one base is affected. So there's a certain point on the line where there's going to be a change in the sequence. So what you were looking at was two normal strands for the cow and for the human. All right, so what they're looking at question number 4 is suppose we make a change. Suppose the person, the first triplet, now on your worksheet, excuse me, so you're going to have to come back up here and look at our first one, which is CCA. And so instead of CCA, the first triplet gene coding for insulin is TAT instead of CCA. All right. Determine what amino acids you would get. So in that box, somebody work it out and tell me what are you going to get instead of the one you got. All right, so again, we've already worked out what it should look at. We already know that when we do the normal one, which was CCA, it translated to GU, and we got glycine. What are we going to translate TAT? First you got to translate TAT, and then you got to find the amino acid that it's going to make on the chart. So tell me what that is. And that's what you're going to write in the box. So I need two things in the box. I need the RNA sequence, and then I need the amino acid that you're changing it to. And everybody should be working on this, not just one person. TAT is going to become what? And this is RNA. Come on, folks. Talk to me, talk to me. Because again, by the time we finish with this tomorrow, all you're going to have to do is submit this worksheet. There'll be no late worksheets because you're doing it with me. Everybody's present. What is TAT? Okay. Thank you, Imani. I'm going to give Imani some bonus points. Professor, what happened to you today? All right, she said it's AUA. So this is what you should have in the box. You need to first tell me what you're translating it to, and then you're going to go and tell me what the amino acid is, which is ILE. All right. So notice that's different from the glycine that it's supposed to make. All right, second, Professor. Okay, Professor, we need to make sure you get some breakfast. All right, question number 5 says, yeah, that's true. That was yesterday for me. Silent mutation occurs when the nucleotide changes, but the resulting amino acids are the same. The protein made this new DNA has no functional difference from the original. What if the mutation occurred in the human gene was the first triplet was changed to GCG? What would be is this a silent mutation? So what you have to do is translate. So remember, the first one was CCA, and we got glycine. So now we're going to say the first one is CCG. What are we going to get? How are we going to, first of all, how are we going to translate CCG? And then what amino acid are we going to get? So again, the normal one is CCA, and we got glycine from CCA. What are we going to translate CCG to, and what amino acid will we get? And is this going to make a difference or not? All right, we're running low on time, and I want to get to sickle cell, so I'm going to tell you. All right, you should get GGC when you translate CCG. It's going to be GGC, and then when I look on the chart for GGC, I'm still going to get glycine. So this is a silent mutation in that it didn't cause a change in the amino acid type. Okay, so the first one did change the amino acid. That one, so that was a point mutation, and it changed the amino acid that you got. Oh, we forgot to say yes or no up here. So will this person be diabetic? Yes, they will, because the code is changed. It's making a totally different amino acid. Number 5, the code is not going to change because we're still making glycine. So they will not have diabetes. Okay. All right. I'm going to start this one. We're going to pick up on sickle cell anemia tomorrow. But for sickle cell anemia, where a person has your blood, the little round ones that look like donuts, that's the way your red blood cell is supposed to look. People with sickle cell anemia have elongated ones. And the problem with that is surface area. The design of the donut-shaped one is designed to carry, the function of a red blood cell is to carry oxygen. And when it's not shaped properly, it doesn't carry enough oxygen. So people with sickle cell typically run out of energy. Some of them you'll see with oxygen tanks because they don't have enough, they can't carry enough oxygen because their red blood cells are not shaped to do so. Okay, so what you have to do, and which I'd like you to have done by tomorrow, is just do the code for both of these, like we did for the cow and the human. Except this time we're looking at a hemoglobin. So that's what the H stands for. There's a normal hemoglobin, which is A, and abnormal hemoglobin, which is which is S. All right. Okay, so we're going to stop there, but you're going to have that figured out by tomorrow. So we start here and be able to answer this question. What type? In other words, is it going to be a point mutation or a silent mutation? That's what we want to know by tomorrow. All right. See y'all tomorrow.